Welcome back. This is the second to last thing. This is the last keynote. I have the pleasure of introducing Danielle Bassett as the final keynote speaker for CCN. Dani is the Eduardo Glant Faculty Fellow and Associate Professor at the University of Pennsylvania. Using her training and background in physics, Dani has changed the field of neuroscience for her groundbreaking work in analyzing connectivity in the human brain and relating this to our understanding of cognition. She applies the tools of network science to many different mental processes in the brain, and in particular has pioneered the concept of small worlds in brain networks. Dani has received a long list of awards, as you might be aware. She is the youngest person who have received the MacArthur Genius Fellowship. She was named an American Psychological Society Rising Star, and she has also received the Erdos Renyi Prize for her contributions to understanding the network architecture of the human brain. Last but not least, I know Dani as a fellow organizer of the conference, and she is a humble and really great person to talk to. So please help me welcome Dani Bassett to the stage.
Thank you very much for that kind introduction. So given the fact that I am the last talk of this wonderful but longer conference, I wanted to give you a relatively broad conversational piece rather than a walk through fine details of data of one particular study. So over the next 40 minutes, I'm going to sort of think about ideas a little bit more than sending lots of data. And I'll give you the citations for the papers so that if you want to see the data, you can do that afterwards. So the title that I gave this talk is Brain Networks That Learn and the Networks That They Learn. And in order for us to understand learning in general, we need to first understand what knowledge is. And for that, I really love this quote from John Dewey in his Democracy and Education. Knowledge is a perception of those connections of an object which determines its applicability in any given situation. Thus, we get at a new event indirectly instead of immediately by invention or ingenuity or resourcefulness. An ideally perfect knowledge would represent such a network of interconnections that any past experience would offer a point of advantage from which to get at the problem presented in a new experience. I love this quote because it illustrates the idea that knowledge itself is a network and has a network architecture. So how do we gain this knowledge network? I think there are many answers to that question. The two that come to my mind initially are curiosity. We can gain a knowledge network by curiosity, and here again is Dewey. Curiosity is not an accidental, isolated possession. It is a necessary consequence of the fact that an experience is a moving, changing thing involving all kinds of connections with other things. Curiosity is but the tendency to make these conditions perceptible. And highlighting the fact that knowledge is an interconnected network architecture. But in addition to curiosity as a way of gathering network knowledge, we can also gather that by example. And to illustrate that idea of how we can gather this knowledge network by example, I'm going to give you an example. And this is from Robert Macfarlane's book called Landmarks, where he talks about Roger Deakin, who was one of his mentors. And he loved Roger so much that he invited him to Cambridge, the University of Cambridge in England, to give a talk on his work. Roger Deakin was an English writer and documentary maker on UK waterways. And Robert Macfarlane remembers back to this time when he invited his wonderful mentor to Cambridge, and he says, "I stared dedicatedly at my shoes, embarrassed that my friend was failing to perform in front of my academic peers. It was only later that I realized it wasn't a failure to perform, but a refusal to conform. Cambridge seminars expect rigor and logic from their speakers, a braced subtlety of exposition and explanation, tested proofs of cause and consequence. But water doesn't do rigor in that sense, and neither did Roger, though his writing was often magnificently precise in its poetry. For Roger, water flowed fast and wildly through culture. It was protean, it was slipshape, and so that was how he followed it, slipshod and shipshape at once, moving from a word here to an idea there, pursuing water's influence too fast for his notes or audience to keep up with, joining his watery subjects by means of an invisible network of tunnels and drains. And this quote, I think, gives you the idea that it's actually very possible for somebody to show their information and the types of the subject that they actually follow through the nature of their lecture. So that made me think, oh, well, wouldn't it be interesting if a lecture could be thought of as a walk through a network? So if that were the case, and if it was a good idea, then perhaps if there was a lecture on linear algebra, it may have lots of ordered structure in it, for example. If there was a lecture on UK waterways, maybe there was a lot more holey or gappy architecture. And if there was a lecture on history, I sort of focus on the top left here, perhaps that lecture would have a more linear form, consistent with the temporal constraints of history and the ways in which that information travels through time. So that makes me ask the question, what is the network structure of different areas of the knowledge space? And is there a good or useful, or if we're feeling particularly positive here, maybe even optimal way of walking through the network in lectures or books or papers, etc.? So I decided that we should ask that question a little bit more formally. Let's suppose that I have 15 ideas that I would like to translate to an undergraduate class at Penn, for example. And those 15 ideas are related to one another in some heterogeneous manner, maybe a little bit like this. Here are my 15 ideas and their interconnections with one another. But I'm challenged by the fact that I need to translate that information linearly, because time is one-dimensional and unidirectional, so I can only say one word in front of the other. So how do I take a potentially high-dimensional abstract architecture of knowledge or of ideas and map it into the one dimension of time in order for somebody to understand it? So that's this problem right here, right? So here is the brain of the speaker or the writer. Here is the network architecture of the ideas that need to be translated. We need to map those into the one dimension of time if we're just using words, so that the person on the other side, here's the brain of the listener or reader, can reconstruct optimally what that architecture actually was. And this is difficult, and we can all probably think back to our undergraduate days and remember professors who were particularly good at this mapping, and then professors who were particularly poor at this mapping. But what we would like to do is to ask if we can make those ideas and those sort of intuitions that all of us may have even as undergraduates, precise and quantifiable and testable in human subjects. So is it possible for us to measure the perception of network topology in a continuous stream of stimuli? That's the challenge that we want to conquer here. So let each of these specific stimuli be a word or an image or a movement, and they will be nodes inside of this graph. Then we're going to let each edge in the graph indicate an allowable transition between nodes. Then we are going to choose a k-regular graph so that local transition probabilities are flat. That becomes important when you want to demonstrably say that it's the network architecture that people are learning and not differences in transition probabilities between ideas or anything else. And then finally, we're going to randomly assign stimuli to nodes so that the graph architecture is the only salient structure here. So just to give you an illustration of what that's going to look like, we're going to construct a sequence of stimuli like this by a random walk on this graph. Obviously, we could take all kinds of walks if we wanted. We could take an Eulerian walk or a Hamiltonian or some other sort of biased random walk. But for right now, I'm just going to show you what happens when we take a random walk through these stimuli. And in order for us to understand whether humans are actually learning this graph architecture, what we do is that at each stimuli, we require the participant to perform a particular task so that their time to react can be used as a measure of how well that edge in the graph was actually learned. So let me give you an example of one of these experiments. So here is a motor version of that type of experiment. So on the left-hand side, you see the stream of stimuli. One set of stimuli is these 5 boxes here, which encode the 5 fingers on your right hand. And there's one of the boxes that is highlighted. That's the 5th finger. So that means press the 5th finger, now press 1 and 3, now press 3 and 4, now press 5, now press 2. This is how your hand sits on the keyboard. And this stream of stimuli is taken as a random, or taken from a random walk on that graph that I just showed you. Now, what's really interesting about this is that when we look at the reaction times on this type of task after people have performed this task for about 35 minutes, you can see that they are actually fairly swift at any transitions that are located inside of one of these modules in the graph, but they're actually significantly slower at this cross-module edge here. And that is relatively unexpected slowing because like I said, all of the transition probabilities are flat. So what do I mean by that? What I mean is that every node in this graph has exactly 4 edges. So that means that when you're sitting at any particular node, if you were optimally learning the data, then you would predict that 25% of the time you would go here, 25% of the time here, 25% here, and 25% here, right? So given that this is a K4 regular graph, the transition probabilities are equated, which means that the reaction time should be equivalent across the entire network. But they're not, which is really interesting. And that's something that is specific, it seems, to humans anyway so far. But we can also do this not just in a motor context, but we can also do it with visual stimuli. So let's take this to be fractal images now. And at each image, we ask whether the image is straight up and down or rotated. And our participants can answer that because we've trained them first on the right way up of each image. And interestingly, this one in particular, we actually told people that each of these fractal images was an avatar for a human, suggesting that they're learning the social network architecture of a community. So you could probably all also think back to cases where you entered a new social situation. It was maybe a class, it was a bar, it was a conference, it was a dorm for undergraduates. And some individuals are particularly good at picking out the structure of social networks very quickly, given the interactions between individuals. And other people are very poor at that. I'm one of the ones that's really, really bad at that. So what we find when individuals are presented with this information is that this surprisal effect, so that unexpected slowing at these cross-cluster boundaries, is actually significantly correlated with their preferences for making decisions in a more individualistic versus collectivistic manner. And in fact, the individuals who showed the greatest surprisal effect were those that tended to make suggest that they made more collectivistic decisions. So this suggests that the type of learning may be important for how we actually understand social networks, not just how we understand motor learning. So those 2 examples are just to show you that actually across several different studies where we change the stimuli themselves, we change the cover story that we present to people, there's a very consistent effect that individuals are much slower at reacting to these edges that cross cluster boundaries, despite the fact that every node has exactly 4 edges. The other effect that we find really consistently is that the reaction times on random walks that go through this modular graph are actually much faster than the reaction times that go through a lattice graph. So both of these facts suggest that humans can perceive higher order network structure in continuous streams of stimuli. And they perceive that in a way that's difficult to understand because we would have anticipated that all of their reaction times would be completely flat on both of these. So what are people thinking? We've been wrestling with this for a while now, and I think our current hypothesis is that as each person stands at a particular stimuli, and they're presented with a particular stimuli, in order to remember which stimulus is likely to come next, so learning the transition probabilities of this graph, they have to not only recognize what they're actually sitting at, but remember the stimulus that came just before this, right? In order to remember a connection, you have to have where you are right now and what just happened just before this. So if we do that, it's very possible, and in fact it's very likely, that there's going to be some probability that we will misremember the stimulus that just occurred. Maybe we were distracted, maybe we were thinking about lunch or anything else that's going on in our lives. And therefore, if there is actually that probability of recalling something different than what actually happened, then we can actually call on some interesting principles from free energy, suggesting that the brain might minimize both errors and computational resources to suggest that the probability of remembering something at a given delta t is given by this Boltzmann distribution with an inverse temperature. Now, to give you an intuition about what would happen then, is on the left-hand side here, what I'm showing you is what happens if the beta, so that inverse temperature, is actually 0. Then this person remembers sort of at random, any stimulus that has occurred previously in the last 30 minutes as what occurred just before the current spot. And if they do that, then their prediction about the network architecture is going to look like this, a fully connected graph. Okay? Now, on the right-hand side, we have what happens at beta goes towards infinity. In this case, they perfectly remember exactly what happened before, which means that their predictions about the transition probabilities in this graph will be perfect, and that's what we see here. That also means that their reaction times will be completely equal across all of those edges, and that's not what we see in the humans. And then here is what happens when beta goes towards about 1. So we have a preference for remembering something that happened recently in time, and we are unlikely to remember something that happened much farther back in time. And if we use that as our prediction about individuals' expectations about the transition probabilities, then we get a graph that looks like this, where we have very strong predictions about what happens inside of the modules, and then we have under predictions about the transitions between modules. Now, why is this? This is because when you take a random walk on a modular graph, you end up spending a lot of time inside of a cluster, right? And so if you have a preference for remembering things that happen nearby in time, you're going to strengthen the connections that exist inside of a cluster, and you're going to weaken the connections that exist between clusters. Now, these are data that, or a model that we can fit to the actual data of human observers, and we can identify what temperature is characteristic of each individual person. But what we'd like to do is to use that model to predict what would happen in new data, should it be collected, really sort of more definitively proving that humans have an understanding of the network architecture implicitly in their head. So here what we did is that we had a ring graph. So this is unlike the other two graphs that I showed you so far. And our prediction was, if after being trained on this ring graph, humans actually have an expectation that is consistent with that architecture, then if we violate their expectations, we should be able to see it in changes in reaction time. And specifically, if we violate this architecture with connecting one node to something relatively nearby, so let's connect the white one to the blue, that's a violation because, in fact, the whites only connect to the green, right? So if we connect the white one with the blue, that's a violation. We would expect them to be slower at that. If they're slower, that suggests that they were not anticipating that, and that's then consistent with them having an expectation of this overall architecture. We can also violate the expectations by taking the white node and following it with one of these red ones, and that would be an even bigger violation, right? Because you're crossing the entire structure of the graph. And so we would anticipate that they would be even slower in those cases. Their reaction time would be even lower. So here's the data that we have, and it's consistent with that hypothesis. So here's the change in reaction time as we go from comparing a short violation to no violation. So you can see a significant increase in reaction time when there's a short violation of the network architecture of the graph. And then here, there's also a significant change in reaction time when you see a long violation. And then here's the difference between long and short. And because this is above 0, what that means is that humans are significantly slower when you introduce a long violation in the network architecture than when you introduce a short violation in the network architecture. So this is consistent with the idea that humans are more surprised by stimuli that are farther away topologically on the ring than closer, indicating their implicit perception of the underlying network topology. So we're pushing this set of research ideas forward in a couple different ways. We'd really like to search for some design rules here. So what nodes in the graph are actually easiest to learn, particularly when we look at more complex network architectures? Could we build a network with more of those particular types of nodes to enhance learnability? And one interesting idea is then to take that back to the lecture scenario and say, is there a better way of structuring lectures that makes the network architecture more easily learnable for the students? Second question, what is the optimally learnable graph? Does it differ across humans? Does it have a topology that's common in language, perhaps, or nature? And that's why it's easier for humans to learn? And then can we use the optimally learnable graph to enhance performance in clinical populations with learning deficits? So I'd like to focus on just one of these questions for a minute, and that is the dependence of graph learnability on the specific human who is learning. And motivating the question of individual differences, I'd like to quote Alexander Pope, where he says, "There's some peculiar in each leaf and grain, some unmarked fiber, or some varying vein. Shall only man be taken in the gross? Grant but as many sorts of mind as moss." And interestingly, there's actually 12,000 species of moss, in case you ever wanted to know. And clearly, there are more than 12,000 humans, both man and woman. But I think this general idea is that there's some beautiful individual differences that we should be taking into account when we're studying learnability. So we've been doing that a little bit in humans, particularly using imaging. What we'd like to understand is what brain networks actually might support learning. So the data that I just showed you is trying to understand what is it about the things that we're learning that makes something easy or hard. But we also want to understand what's going on in the brain that allows for all of this to happen. So what features of the brain might support the learning of graphs, and then might differences in these features explain differences in our ability to learn? And that's motivated us to sort of go back and think much more broadly about what network substrates might be important at the whole brain level for adaptation and learning. And if you go back through history, there's a very common refrain, which is that modularity, or separability of components, is extremely important for systems to display in order to adapt and change to an evolving external environment. So we could even go back to Darwin Finch's, where the idea that there's modularity of structure and morphology that allows for evolution. We could also think back to ideas of modularity of mind. I'm particularly thinking about Jerry Fodor's book called Modularity of Mind from the 1980s, although I think the complete independence of components is not exactly what I'm talking about. I'm just talking about large separability, not complete separability. And then there's some really nice work recently in the area of network science that's demonstrated that from data we can extract where these modules actually are in a data-driven manner that's actually relatively robust. So we can ask, motivated by these observations, we can ask the question, are brain networks actually modular at the whole brain level? And then does that modularity help us to understand large-scale signatures of adaptation and learning? So the way that we often do this, for people who are not familiar with this part of the field, is that we'll take a invasive or non-invasive neuroimaging technique. In this case, I'm showing you an MRI machine, but we could use EEG or MEG or electrocorticography. And then we parcellate the brain into between, let's say, 200 and 1,000 different regions. They are usually anatomically or sometimes functionally defined. Then we look at the regional activity time series. And then we'll compute some measure of statistical similarity between those time series. And sometimes that statistical similarity is referred to as functional connectivity. In this particular case, I'm showing you a wavelet coherence matrix, which is between 112 different brain regions. And once we've built that network, that matrix of connectivity, we can look at its underlying architecture. Here, for example, is data that's actually taken with an fMRI when individuals are just sitting, resting inside of the scanner. And what you can see is very, very clear modularity of the network organization. So that answers our question number one. On the left-hand side, what you're seeing is the graph itself. The colors of each of these nodes is the same as the colors on the right-hand side. So when you see this large blue module here, that's the same as these regions here. So in other words, these are visual areas in the visual T-Rex, as we heard at 9 a.m. this morning, for anybody who was there. So clearly modularity is definitely present in these networks, but then the question is that relevant for how these systems adapt and respond in learning settings? So to ask that question, we've tried to push the field a little bit further in accounting for the dimension of time instead of looking at single snapshots of the static network architecture. So we would start similarly. We would start with the brain and our regions of interest. We would again extract the regional time series, but now we'd cut those time series into individual windows, and in each window we would estimate the statistical similarity between the time series. So what that would give us is a set of networks over time. And the sort of naive approach after we've extracted this set of networks that's temporally evolving is to examine the architecture of each of those windows separately. However, that would ignore the fundamental fact that this is a dynamical system that is evolving in time, and therefore what we'd really like to do is to have a network representation that remains true to that fact. So to do that, we move from these independent networks that we can extract to what's called a multi-layer network. So this is an extension of the model in time, where now each window is represented by a network, but then the networks are connected up with what are called interlayer links. And those interlayer links connect a region of the brain in one time window to itself in another time window. So V1 in time window 1 will be connected to V1 in time window 2, which will be connected to V1 in time window 3. So this takes our adjacency matrix that we usually study and can map it out into an adjacency tensor. On that, we can then perform multi-layer modularity maximization, which is a machine learning tool to extract clusters inside of the network as a function of time. Now, over the last couple years, we've been doing this across a wide variety of different tasks, and we have found that there's this really interesting characteristic of the data where the modularity of the networks actually remains relatively steady over time. So none of these modules completely disappear or reappear. However, there is reconfiguration of the network architecture and of the modules, often along the module boundaries or in between the intersections between two modules. And I'm going to show you that schematically up at the top here. So this is a little graph that has three modules, the peach, the yellow, and the blue. I want you to focus on this one node at the very top, which is peach in time window 1, because it has only connections with the peach module. In time window 2, it has a strong connection to the yellow module. In time window 3, it has 2 connections to the yellow module and only 1 to the peach module, which means it's now assigned to yellow. And then on the right-hand side, it has no connections with peach and it only has connections with the yellow module. So that's an illustration of the fact that a node inside of this network can actually change the module that it's affiliated with over time. And we find that this happens very frequently in brain network data, temporally brain network data. It happens very frequently in the regions that tend to do this most often are regions in frontal parietal cortex. Interestingly, individual differences in that characteristic, so the number of times that a brain region switches between modules during a task, is correlated with individual differences in a range of different cognitive functions. So visual motor learning, cognitive flexibility, working memory, so this is particularly an N-back task, reinforcement learning rate, and interestingly, positive mood. So this suggests to us that modularity certainly is present in these data and that it allows for this adaptability, specifically at the intersections between the modules where these regions can bounce back and forth between different modules. So just to give you an illustration of that idea with some data, this is a paper that I saw Marcelo in the audience. Marcelo's here, so he's the first author on this paper that just came out in NeuroImage this year, where we have resting state data that was taken for 5 minutes before 20 participants participated in a 6-week training regimen of a discrete sequence production, so a motor skill learning paradigm. And our hypothesis was that when someone has relatively modular resting organization, that allows for them to be fairly flexible and adapt over long periods of time. Whereas when somebody has more constrained, more sort of interconnected modules, that would have them be much more constrained. It's very difficult for them to change one module independently from another. So that was our hypothesis. So we predicted that these individuals that have more modular organization between these two modules that are important for the task would learn better. So here is the visual motor connectivity. So that's how much the visual module is connected to the motor module. at rest in 5 minutes, and then on the y-axis is the learning rate over the next 6 weeks of practice. And interestingly, what you see is that people who have less visual motor connectivity are those that have better learning over the next 6 weeks of practice. So we think this is really interesting because it supports the underlying idea that modularity is important for adaptive function and can potentially be used for prediction of future behavior even over the course of 6 weeks. Now, one of the interesting questions is, if that's true, is it possible for us to intervene to enhance brain network flexibility in somebody who maybe cannot change certain things about their lifestyle or brain themselves? And for that, we've focused on looking at the underlying drivers of what is producing this brain flexibility at the very, very large scale. Our hypothesis was that flexibility at this large scale could be driven by a change in the excitatory and inhibitory balance in the brain. So here, in order to test that hypothesis, we looked at 41 healthy subjects who were taking dextromethorphan, which is obviously it's a relatively dirty drug, but it is known to affect the NMDA receptor, and specifically it's an NMDA receptor antagonist. So what you see here is that if individuals are on placebo, they have a sort of very normal, for the data sets that we've observed, normal level of flexibility, but on DXM they have heightened flexibility. So that's relatively interesting. It means that potentially this whole brain network reconfiguration is something that can be modulated by a drug that changes the excitatory inhibitory balance in the brain. Now, we can also couple that observation with this one on the right-hand side, where we have flexibility of healthy controls, people with schizophrenia, and relatives of people with schizophrenia. So here you see that the flexibility in people with schizophrenia is significantly higher. That's actually consistent with the hypothesis, one of the hypotheses about the disease, that is associated with a change in the excitatory inhibitory balance in the brain. So that suggests not only that this is something that we can modulate, but also that it could be modulated in psychiatric disorders. Now, hopefully you have a little bit of cognitive dissonance right now, and that was purposeful. And the cognitive dissonance that I'm hoping some of you have is the fact that I think what I've described to you is an inverted U-shaped curve between optimal cognitive function as measured by NBAC working memory performance, cognitive flexibility, and various types of learning, both reinforcement learning and visual motor learning, and functional brain network flexibility. So initially, the first few studies I showed you suggested that these cognitive functions are associated with higher flexibility, but then I showed you on the right-hand side that schizophrenia actually has much higher network flexibility than healthy controls, and that's also associated with weakened performance on a lot of these same tasks. So I think that what's happening is that there's an inverted U-shaped curve between these cognitive functions and network flexibility, and that's something that we're hoping to unpack a little bit more over the next couple years. So that leads me to the final set of vignettes that I wanted to offer to you today, and which are motivated by the question of how might this reconfiguration of functional networks be driven? What perturbative signal is potentially responsible, and how is it propagated? So what we'd really like to do is to have, like Heisenberg, be able to say, one should be able to prove, for example, that the introduction of a small perturbation in the dynamical problem leads to additional terms for the energy of the type of the ones found by Kramers and Born. In other words, wouldn't it be wonderful if we had a perturbative theory of the brain? That would be great, right? All right. So not that we're going to exactly get there, but what I'd like to offer is a direction for research that I think is very relevant. So when we think about cognitive control, that is a cognitive sort of process that we think of as being capable of reconfiguring large-scale brain network organization. Now, we also know that cognitive control, particularly the ability to switch between different task states, is often associated with activation in frontal parietal circuitry. We also think that these regions have very different underlying white matter architecture that potentially enables them to do that switching. And that's supported by some recent data that was led by a study led by Rick Betzel, who some of you have heard earlier on in this conference, in this paper that also just came out this year, where he used weighted stochastic block models to show that these regions in frontal parietal cortex are actually associated with very complex patterns of interconnections that are not just modular and not just hub-like and not just many of these other simple characteristics that many of us have studied before, but are much more diverse and varied. And that's motivated some of the work that we have been pushing forward on over the last couple of years to ask whether cognitive control can be thought of as network control. So network control theory, for those who aren't familiar, is a field subdomain of control theory in engineering. And it asks the question of whether you can take a network architecture and if you perturb one piece of it, can you predict what happens to the rest of the network? And it has developed some really beautiful computational tools and theories for understanding how to do that. Traditionally, it's been applied to mechanical systems and technological systems, not so much to brains. So we asked the question of whether it could be usefully applied to brains, and if that would help us to understand cognitive control as a network-level process. So the idea is that you can inject activity into different brain regions, and you want to be able to predict what's going to happen to all the other brain regions inside of the network. And what you'd like to have is a way of predicting that when you intervene, you're going to push the system to a different place than where it would have gone naturally. So if you're very curious about the theory underlying some of this, there's a paper from Jason Kim, who I also saw in the audience earlier, that just came out this year as well. So if you're curious, that's where I would go for the sort of really basics of the theory. But I'm going to give you an intuition now. So here is, if we want to understand network control, we have to not just understand the organization of the architecture, but we also have to write down a model for the dynamics that occurs on top of that architecture, right? So here's the model of dynamics we've been studying initially. It's a relatively simple, very, very simple actually, linear model of dynamics where we have the state of the brain x at time t+1 is going to be equal to the connectivity, the structural connectivity from white matter, that's your adjacency matrix A, times the state of the brain at time t, and then plus some control, so the control energy U being injected into kappa regions. Now from this, we can write down what's called the t-steps controllability Gramian, and from there we can extract various metrics of how the system responds to exogenous input. One notion that's going to be important for us is what's called average controllability, which is basically the impulse response of the system. It's given by the trace of the inverse of the controllability Gramian. And then the second one is called modal controllability. So for this one, you can actually extract it straight from the adjacency matrix A. So we'll let V sub j be the jth eigenvector of A with a particular eigenvalue lambda j. Then if the element Vij is small, you can intuitively imagine that the jth mode is going to be very poorly controllable from that node I. And given that, you can actually expand across all of the modes and all of the regions to identify which regions of the brain or which nodes inside of the network can most easily control these poorly controllable modes. Now, if we do that in a brain, what we find is that cognitive control areas, so I'm highlighting the regions that we were interested in here in green, cognitive control areas show very high predicted levels of modal controllability. So here on the right-hand side, you can see the amount of controllability, and I've separated it into average controllability and modal. So modal is this ability to control the poorly controllable modes, which means intuitively it's the ease with which these regions can push this system into very distant states on an energy landscape. Now, I find that really interesting because these are also the regions that are important in task switching, which is intuitively similar to changing the state of the brain to something quite different, right? So these regions that have high modal controllability based on their white matter organization are also the same regions that are activated when you have to switch between different tasks. So that's some initial evidence that network control may be thought of or cognitive control might be thought of as network control. There are two other pieces of data that support that notion as well, and you can, if you want to see some of the specifics, I have the references here. So we find that network control grows as children develop cognitive control, and that's shown here on the right-hand side. So here's average controllability and modal controllability, and each data point is a person in the Philadelphia neurodevelopmental cohort. So these are kids between the ages of 8 and 22 years of age. This is a period across which cognitive control capacity changes drastically. So what we find is that average controllability and modal controllability both increase over this age range, with the highest levels being present in the 22-year-olds and the lowest levels being present in the 8-year-old kids. And then in this work from Eli Kornblath, we show that individual differences in network control predict individual differences in executive function in youth. Again, suggesting that cognitive control might be thought of as this very, very broad network control process across the whole brain. And all of that is constrained only by the underlying white matter connections between large-scale brain areas. Now, the last piece of data that I wanted to show you is, let's see if that's true. All of that is correlative, right? Let's see if we can actually intervene in a brain in such a way that's consistent with this theory. So for that, this is preliminary work that's on bioRxiv. You can see the preprint, it's not yet published. But what we've done is that we've taken electrocorticography data from patients with medically refractory epilepsy, and they've all agreed to undergo a stimulation regimen where different regions of different electrodes are stimulated, and then there's continuous recording of what happens throughout the rest of the brain. And our hypothesis was that if we stimulate regions that are high in modal controllability, we will be able to support executive function. And that is consistent, even though it's a small sample at the moment, it's consistent with the data that we have. So here's modal controllability of the region that's being stimulated on the x-axis, and on the y-axis is a change in the memory encoding state probability. And you see a positive correlation here, suggesting that stimulation to these modal control regions is consistent with pushing the system to better memory encoding states. So where we're taking this last effort is searching for optimal stimulation. Network control has implications not just for cognitive control or understanding intrinsic control capacities of the brain, but also understanding exogenous control. And that's important for both open loop and closed loop paradigms. What we're doing is that we're trying to validate the utility of this control theory in predicting power changes in response to grid stimulation in ECOG data. We are trying to understand the different time scales of control and the frequency specificity of that control. And then third, we're trying to determine the relative role of white matter connectivity versus something else, like effective connectivity or something that's functionally or context dependent in predicting the effects of stimulation. Fundamentally, what we're really hoping to do is to have a theory for how both intrinsic and exogenous control of brain-wide network states is possible. So with that, I'd like to sort of summarize what we've done so far. So we started with John Dewey, wonderful educator and philosopher, and then we walked around in knowledge network learning for a while. We took a small hiatus across Alexander Pope, and then went through brain network dynamics. Then we had a transition along Werner Heisenberg, a wonderful physicist, and then ended in brain network control. Now, there are a couple of things that need to be done in order for this to be convincing. You can probably all list many, but here's a few. So number one, I didn't take a random walk, despite the fact that I told you that all of our studies take random walks. So you could probably imagine that random walks as a model of a lecture is probably not ideal. Otherwise, maybe I would have chosen that. We also need to link John Dewey with brain network control. So notice that there was a disconnect here. I still haven't figured out how to link Dewey with brain network control. So that's for me to do. And then I also think that we could potentially pull in some ideas from algebraic topology to fill in this central cavity, to understand not just each of these pieces as independent units or even connected in a line, but to understand how they can mutually inform one another in a broader research direction. So with that, I'd like to acknowledge the foundations and institutions that have supported the work. I also wanted to particularly highlight a lot of the individuals who are past trainees that did much of this work but are now faculty. I saw Shigu in the audience. Many of these individuals were important in the initial stages of a lot of this work. We also have 3 past trainees that are just brand new faculty members starting this month. And then 2 faculty collaborators that I didn't get a chance to mention earlier. So Ted Satterthwaite and Raquel Gurr were extremely important in allowing us to have access to the Philadelphia neurodevelopmental cohort data that was that we used to understand network development in kids. So with that, I'd love to see if you have any questions.
Hi, a great talk as always. The question I have is about the control elements of network control. Because of course, if we're doing smaller scale neuroelectrical recordings, you know that there's different kind of ensembles within a region that have different downstream circuitry, right? So you might imagine that one input to that area like V1 has very different consequences for say MT or for an area like higher order visual areas than another cell assembly. So when you think about this control problem, it seems like you're almost saying like if you just like douse with micro stimulation the whole area, what would happen? How kind of plausible is that in the framework of what we think the brain actually does in its natural control problem?
Yeah, that's a really great question. So I should say that all of this work is only going to be a first order approximation, and again, at the very, very large scale. So I'm not suggesting that this is going to be a fine scale approximation at the small scale. But what we'd like to understand, I think what motivates a lot of the problems that we're studying is the brain has very interesting heterogeneous architecture at this large scale that must play a role in constraining the first order dynamics. And that's what we're really trying to understand at the moment. We would like to take this to the neuronal scale as well. There's some really nice work from Emma Towson and Petra Vertes and Laszlo Barabasi using this to understand C. elegans motor dynamics. So it is possible to study it at the small scale as well. But I agree that if you're going to be studying particular circuitry and particular inputs, that we're going to need to expand the model of the dynamics that I wrote down. So the structural connectivity might stay the same, but that model of the dynamics is a simple diffusion, right? And so that's only going to be relevant in a very broad way. It's also linear rather than nonlinear, and so some of the processes that you're imagining have many nonlinearities that will be important. Now there is some work. I mean, nonlinear control is hard, but there is some nice work that's been done recently in extending that into the network context. And so I think the stage is ripe for pulling some of that theory over to neuroscience. Yeah.
Danny, great talk as always. I'm having trouble reconciling two parts of what you presented. So you have the white matter topology identifying regions that are kind of critical for this modular control. And then you kind of show the adolescent work where you kind of show that control changes over development. But the topology of a lot of those big fascicles is pretty established pretty young. I mean, the myelination might change between edges, but the topology is established very early on. So is there something else about the network that's developing across time that's leading to this change in control, or do you think these are just a hodgepodge of different systems and you're picking up different aspects of the system?
That's a good question. I mean, we have Graham Baum is a graduate student with Ted Satterthwaite who has a nice paper in Current Biology this year where he shows that the network architecture of white matter does change over this age range from 8 years to 22 years. It increases in modularity, but then it also has a strengthening of edges that are like right across the boundary of the modules. So I think there are some, yes, the overall architecture is there, but there's fine tuning that happens between 8 and 22 years that I think we're picking up on in the control analysis.
Thanks.
Sorry.
Hi, thank you for the wonderful talk. I have a question about the knowledge network learning aspect. It seems by putting it as a graph that you're basically supposing that it makes sense to traverse the edges of the graph as you deliver the knowledge to some audience. Is it possible that it actually might be relevant to make hops sometime, and that you actually don't want to fully traverse the graph as you hope that your audience can better reconstruct it, and that essentially the ideal model is not necessarily a random walk of some sort, but includes transitions that are illegal but helpful in understanding?
I really love that question. I don't think we want a random walk. I also don't think we want a fully ordered walk. I think we want to have some jumps in the space. And that's motivated by this paper that just came out yesterday, which I'm super excited about, from Ann Sizemore, who's a graduate student in the lab, where she studied language acquisition in toddlers. And she shows that, in fact, the acquisition of these words is best characterized by not a gradual sort of wave-like dynamics increasing the understanding of words that are related to one another. It's not crystalline in a graph structure, for example, but there are these big gaps in knowledge. So there's a word here that's really unrelated to almost any other word that's in the vocabulary, and then slowly those gaps fill. We're also seeing that in textbooks, actually. So we're doing semantic analysis of textbooks, and we can see that it's not a steady wave-like dynamics or a crystalline structure of the graph. It's also not a random walk. There's this combination of local growing and then really far reaches. And I think that that's important for how we, in general, grow the knowledge space that we have. So now whether or not that's going to be useful for lectures, I think probably yes, but I think that needs to be shown quantitatively. So we're doing some work in classrooms now to see if we can show that.
Awesome, thank you.
Really amazing talk. Definitely a lot to think about. One of the things that I feel kind of, I guess, on my own confused about is how, like, as the brain network is changing over time, the node definitions are changing over time as well. Like, are there methods in graph theory that can take that into consideration, or like at least account for something like that?
Yeah, that's a great question. Absolutely, that's true. So we have one of the simplifications is that the nodes stay the same through time, but you're right that functionally that's not necessarily true. So what you can do is when you build a multi-layer graph, instead of having a single interlayer link between one node in one slice and that same node in the next slice, you can have multiple connections. So this node has connections with all of these new ones. It's sort of partially this one, partially that one, partially that one. And so you can draw those interlayer links and have them multiplicitous instead of singular.
Thank you.
That's the best way, I think.
Quick question. How stable are those networks across different conditions? Stimulus conditions, task conditions, whatever behavior you engage in?
The functional ones, sorry, I'm losing my voice. The functional ones are, it depends a little bit. There's certainly a lot of the modular architecture that remains the same across various task conditions and the rest conditions. What we actually see in the data that we've been studying from the Human Connectome project, for example, is that the interconnections between modules often strengthen with tasks. And at rest, there seems to be more segregated modular architecture.
And if they strengthen in one task, are they also going to strengthen another task?
It's a little bit, some of it is consistent across tasks and others are really specific to which regions are activated by that task. So there is task specificity and task general processes as well. So in other words, the answer is complicated, but specific to the task.
So my big question, I guess, is whenever I see one of these networks, I wonder, should I remember it? Or is it going to be different in the next talk?
No, the general modular structure from the resting state graph that I showed you is very consistent over many labs, many data sets, many people. That's very consistent. That's not going to change. All right. Thank you.